Good morning, Bio 218. Here is that promised agglutination video. So we're going to start with some basic terms from immunology. We're going to start with B cells. And so B cells are a type of lymphocyte. And we learned during the blood lab that those B lymphocytes are rarely in the bloodstream. They're much more likely to be in the lymphatic system, most likely the lymph nodes. So B cells are the cells that are responsible for making antibodies, and they don't make them directly, but that's all you need to know right now is that B cells make antibodies. And you need to know that antibodies prevent invaders from either attaching to or getting into body cells. And after those antibodies prevent invasion, those invaders are going to be eaten by phagocytes. So any of those cells, those macrophages or neutrophils, those types of cells that eat other cells. So now that we have some terminology down, we're going to talk about how antibodies prevent invaders from attaching to or getting into body cells. And the way that they do that is by either grabbing onto the attachment sites or making big clumps of invaders so they're too big to get into the cells. So, and the process that they use is called agglutination. So agglutination is attaching to antigens on an invader's cell surface. So here we have some bacteria, and on the surface of the bacterial cells are these antigens. Okay? These antigens tell our body that the cell is not us. Our cells also have markers on the surface, but those markers tell our immune system this cell is part of you. Okay? So these antigens are kind of a warning system. They let our body know that a foreign invader has shown up. And our body is going to learn how to make antibodies for these foreign invaders. So antibodies are specific to a specific antigen type. So this antibody right here can only attach to this triangle type antigen. And this antibody over here can attach to this rounded antigen. But they have many attachment sites. So this antibody is attached to this bacterium, and then this antibody is attached to this bacterium and this bacterium. And what's forming here is a clump, much like the clumps that you saw when you did the Staphylococcus tests last week. So again, these sites, I'm going to magnify and show you, are these sites, these antigen binding sites. And these antigen binding sites are specific to a part of the antigen. Okay, so the antibodies are not just going to bind willy-nilly to whatever comes in, just cells that have that specific type of antigen. Okay, so when we were in lab, we used Staph Tex latex beads, and they're coated with IgG. IgG is a name for a type of antibody, and that IgG recognizes protein A, free coagulase, and bound coagulase. And these were things you should have identified doing the pre-lab assignment. But for now, we're just going to say that they are antigens found on the outside, or found on the outside of the cell, or within the cell. So protein A, free coagulase, and bound coagulase are all found on Staphylococcus aureus. Staphylococcus aureus is a nasty thing. It causes pneumonia, it causes hard to treat skin infections, including MRSA. Staphylococcus aureus is closely related to Staphylococcus epidermidis. But Staphylococcus epidermidis is less virulent. It's less likely to cause illness. It's covering our skin right now. All of us have it on our skin. The only time it becomes trouble is if you have any plastic that's put into your body, say like an artificial valve or what have you. It can create a biofilm on plastics that can sometimes cause trouble. But for the most part, Staphylococcus epidermidis and humans get along just fine. It's important to know if you are dealing with a Staphylococcus aureus based infection then. And so there's this really nice thing that Staphylococcus aureus is what has the protein A and the free and bound coagulase, and Staphylococcus epidermidis does not have protein A and does not have coagulase. So we can determine which of these strains we're looking at by whether or not it clots or clumps when we mix antibodies for protein A and coagulase. And I just wanted to mention, while these pictures show them in different colors, those are kind of computer enhanced colors. There's no actual color when we're looking at staph infections. And so if only they were purple and green, that would be much easier, but they're really pretty clear. So that's just kind of going over the Staph-Tex activity again. So the Staph-Tex latex beads are coated with antibodies that recognize protein A, free coagulase, and bound coagulase. So if you mix the Staph-Tex beads with Staphylococcus aureus, we're going to see clumping. And if we mix it with Staphylococcus epidermidis, we're not going to see clumping because those antibodies cannot attach to anything on epidermidis, but they can to aureus. So you should have had, on the Staphylococcus aureus sample, you should have had a positive result or clumping. And on the Staphylococcus epidermidis, you should have had a negative result, no clumping. Next, we're going to talk about blood typing. And blood typing is another situation where we're using agglutination to identify the presence of antigens. So if you see clotting, it's telling you you have a positive result. So type O positive, type O blood does not have any A antigen. So when you mix it with anti-A, no clumping occurs. When you mix it with anti-B, no clumping occurs. But this is O positive, and so when you mix it with anti-D, clumping occurs. And our control here is a negative control. If we saw clumping in our control, then we wouldn't be able to trust it because it's a negative control. There's no clumping, so we're good. I'm going to hop down to A positive. So type A blood has A antigen. So when you mix anti-A with type A blood, you see clumping. When you mix anti-B with A blood, no clumping. And when you mix anti-D, which tells you about the Rh factor, with this, we'll go down to this A negative blood, you see no clumping. So if it clumps, it's a positive test. And if it doesn't clump, it's a negative test. So negative, negative, positive, positive, negative, negative, positive, positive, positive. All right. So we've kind of gone through these. You can spend some more time with these tables on your own, but I just want to look at a chart that explains the antigens and antibodies that each blood type makes. So if you have type A blood, that means that on your blood cells, on your red blood cells, is type A antigen. Okay? And so when you mix antibodies for A antigen in type A blood, it's going to clump. A person with type A blood is going to make type B antibodies. That is why we cannot give somebody a transfusion of type B blood if they have type A blood, because their bodies make antibodies for type B blood. So if you have type A blood, you can get a transfusion with type A or type O. We'll explain why shortly. If you have type B blood, you have B antigens on your red blood cells, and you make A antibodies. So you can't get type A in a transfusion because you'd have a reaction, a clotting or a clumping reaction. If you have type AB blood, you make both type A and type B antigens. You don't make any AB antibodies. Sorry about that. And so you can get transfusions of A blood, B blood, AB blood, or O blood. If you have type O blood, that means that you do not have any AB antigens. And having no AB antigens means that you make antibodies for type A and type B. So AB is seen as foreign. Sorry, I keep going back and forth here. And so your only transfusion option is type O. Okay, so if you have type O blood, you can give your blood to everyone, but you can only get type O. And then the next thing we're going to talk about is Rh factor. So if somebody is Rh positive, that means that they have Rh antigen. I'm going to make this a little h because that's the right way to do it. So they are Rh positive, they have that Rh antigen. They're not going to make any Rh antibodies. And so they can get either Rh positive blood or Rh negative blood. If you are negative, though, that means that you have no Rh antigen. But here, people that are Rh negative don't automatically make Rh antibodies, only with exposure. So transfusions can only come from Rh negative people, or the body will start making those antibodies. Where this really comes into play, though, is pregnancy. Pregnancy is, so if somebody is Rh negative and their partner is Rh positive, their baby is going to potentially be Rh positive. During the first pregnancy, with no treatment, a person will go through the pregnancy, deliver, and often during delivery there's maternal fetal blood contamination or exposure. And that is the point when the mom's body starts, it's likely to make Rh antibodies. And so that first baby is fine, but if a second baby is Rh positive, that mom's body is already going to be sensitized to Rh, and the mom's body may start attacking the red blood cells of the baby. And so babies with Rh incompatibility, they're born, they're usually low birth weight and often anemic. So that's with no treatment. But fortunately, we live in the days of modern medicine. And so if you are Rh negative and pregnant with and pregnant, they don't even go into testing, because testing that type of testing for blood type is actually a little risky. What they will do is about halfway through the pregnancy, you're going to get a RhoGAM shot. And RhoGAM is actually Rh antibodies. And having the RhoGAM shot kills any Rh positive cells that have made it into your bloodstream. Okay? And what the goal is to prevent your body from learning how to make Rh antibodies. And so you're going to probably have 1 or 2 more shots during your pregnancy and shortly after delivery, so in the hopes that your body will never learn how to make those Rh antibodies. As long as you keep administering Rh antibodies, those cells will be destroyed before your body learns about them. So again, Rh antibodies are only made with exposure. And if you are Rh positive, you can get positive or negative blood, but if you are Rh negative, you can only get negative blood. I strongly encourage you to do the activity that is on Moodle to further help your understanding in ABO blood groups. I will see you next week. Bye-bye.